Hello, everyone, and welcome to the Analysis of Mitigating Concrete Cracks with Bacteria webinar. My name is Paula Hyman from the Ohio LTAP Center, and I'm going to be your technical support for today's webinar. Just a couple of housekeeping items. If you have questions throughout this presentation, I ask that you please type them into the questions box, and our presenter is going to address your questions at the end of the presentation. This webinar is being recorded. Providing that the recording is successful, the YouTube link and a copy of the presentation will be emailed to all participants and uploaded on our LTAP website. I want to thank you in advance, and with that, I'll pass things off to Ms. Lisa Burris, Assistant Professor of Civil Engineering with The Ohio State University. Lisa.
Oh, thanks, Paula. Hi, everyone, and thanks so much for coming to learn a little bit more about using bacteria in concrete. As Paula mentioned, I'm an assistant professor at The Ohio State University right now. I've been there since about 2017. And I also want to thank, this is a pretty collaborative effort, as you might guess. Like I assume most of you, I didn't know much about bacteria when this started, hadn't worked with biological systems, just spent a lot of time thinking about concrete and hydration and sort of inorganic chemistry things, not about living things. And so we have also Professor Natalie Hull, who's also at The Ohio State University, who is an expert in biology and DNA-based understanding of bacterial systems. She's done a lot of work with COVID wastewater project and things like that. We also incorporated one of my friends who works in Istanbul, but has her PhD was in bacterial concrete systems, Zeynep Basaran. And then we had 3 graduate students assisting with the project, Cansu Akraturk, who just finished her PhD this summer, and Judith Strayhoff, who finished her master's last year and is now working in practice, and Yijing Liu, who is currently working on her PhD, specifically looking at bacterial systems, not in concrete. I'm going to turn off my webcam now, as I've been told it can kind of make things lag, and then we'll get started. I did also want to say just a thank you to ODOT and the oral group who put together this project idea. It's very forward thinking. Something that's like a really interesting science question, but isn't usually something that DOTs and governmental transportation agencies would think to fund. But I think it's really essential. There's been a lot of work done in this field in the past decade or so on using bacteria in concrete, but all of it has been done at a very, very small scale. The scale that you can usually get the very science-focused agencies like National Science Foundation to fund at is usually focused more on things that are a couple inches in size and really understanding the science, whereas what we need now is information on how to scale this technology and implement it well in the field. And so I hope that based on this research, we'll see a path forward and some additional questions that maybe can be answered in the future through additional funding for large-scale work. But it was really innovative for ORL to fund this. So thank you to everyone who thought about doing that. So the agenda for today's talk, we're going to start with some background and objectives about what the system is, how it works, what it looks like, what types of bacteria, those kinds of things. And we'll talk about the work that we did to optimize the production process. Then we're going to talk about moving into cementitious systems, mortar and concrete performance testing, including the, as far as I know, first ever field scale implementation of bacterial concrete. And then the last part of the project was to assess how much this actually costs and if it's worth it from a durability perspective. And we'll end the presentation with my recommendations for moving forward with this system. So first, a little bit about biomineralization. So this is a natural process. If you've heard of people taking biomimetic approaches to different science ideas, there's some of this going on in structural engineering, there's some going on in additive manufacturing and 3D printing kinds of fields. And we're using that same principle here to help densify and heal our concrete. Microbial induced calcium carbonate precipitation, or MICP, which is also in kind of shorthand referred to as biomineralization, is something that's done, and you can see it happening in corals. So these are bacteria and other organisms that consume one product and then secrete a calcium carbonate, just like a limestone, to build up a structure. And so the idea is that we'll use these organisms to fill cracks once they happen in the concrete, or we may be also able to use them to densify the concrete, remove at least surface porosity, and reduce the amount of infiltration of things like chlorides or sulfates into our concrete matrix, thereby increasing the durability. There's two ways that this can happen. The first is urolytic activity. So we essentially take urea, the bacteria react with it by secreting this urease material, and that creates an ammonium compound that will combine with carbonate and calcium that's in the system to form calcium carbonate. The other alternative way is an enzymatic activity. So this is the creation of a protein by the bacteria that will split up. So it pulls calcium or pulls CO2 from the air, and then that can react with calcium from the environment to form calcium carbonate. Both methods work to produce the calcium carbonate product that we want in the end, and both have been shown to reduce hairline cracks in concrete and reduce permeability. Okay. So our objectives in this study were first to understand requirements to upscale this system to take it from small lab specimen size to real application size. We wanted to assess the feasibility of doing that, and also if we could achieve the benefits that are supposed to accompany biomineralization in larger scale mixes in the real environment. We also specifically wanted to look at use of local Ohio materials for these systems, so that we're not shipping things in from far away and increasing cost further. So specifically, in order to meet those goals, we wanted to look at what the best bacteria is and what's available in Ohio, what growth media to use to grow the bacteria and feed the bacteria so it will precipitate calcium carbonate. We wanted to determine the overall effect the bacteria would have on things like strength and sorptivity and porosity of the concrete. And we also wanted to see if we could use similar concrete mixes as we are already using in Ohio. So things like a QC1 DOT mix and typical admixtures. And even we also investigated the use of calcium sulfoaluminate cement as well. We also wanted to understand curing of these systems and how that affects the ability of the bacteria to live over time. And specifically, we wanted to investigate systems that we thought would be a little bit easier to implement. The only used curing method to date has been ponding cubes in a water bath, or not a water bath, but a nutrient solution bath. And obviously, we're not going to be using hundreds of gallons of curing solution on top of a roadway in order to simulate the bacterial precipitation. So we wanted to look at things that we thought could be a little bit more possible to do in the field. We also did want to investigate the effectiveness of the bacteria at stopping or reducing hairline cracks. And we wanted to understand the effect of using this material on the cost of our applications. Okay, so there are 4 phases of this project. The first was to select microorganisms and figure out how to grow them. Then after we looked at optimizing variables like pH, temperature, the concentration of the solutions used, the amount of time it takes to grow things, and we also investigated several different sources of bacteria in order to try and find the best one for the system. We then took that to the concrete lab and cast mortars and concrete and evaluated the effect of different variables like use of fly ash, use of lightweight aggregate, how to cure the material, and the impact of environmental conditions like varying temperatures or salt application. We then placed a larger scale concrete installation, which I'll show you a picture of later, and then finally tried to assess the impact of life cycle costs and durability. The overall goal of this work is to develop a more resilient concrete system, something that can correct some of the inevitable concrete cracking that we experience, and just improve our durability through use of microorganisms. So the first step was to identify bacterial sources. There are 4 things that we need to have from a type of bacteria. Not all bacteria on Earth biomineralize, and not all of them will be suited to use in concrete. So we need a very specific set of traits. Those include it should be able to biomineralize and secrete calcium carbonate. Otherwise, what's the point? We also need to be able to grow it to very high concentrations so that we have enough bacteria in the concrete to do the job that we want it to do. We also need bacteria that's endospore forming. I've shown a picture on the bottom of this slide that kind of illustrates the differences in traits of the bacteria. So we have two states in endospore forming bacteria. One is called the vegetative state, and despite the name vegetative, that's actually where the bacteria is active and it's eating food and it's secreting calcium carbonate. So we need something that can be alive and growing and secreting, biomineralizing. But then we also need it to go into spore form when the conditions are not ideal. And most of the time in our concrete, the conditions are very not ideal. We have high pH, we have lack of nutrients and water sometimes if we've self-desiccated the concrete. And so we need the bacteria to protect itself during those lean times and then be able to regen into the vegetative state when we supply it with water and nutrients, which could even happen if we have a crack and then it rains. So we must have an endospore forming bacteria, and then we also need something that's resilient to high pH since we have such severe high pH conditions within the concrete, usually around 13.5 to pH 14. So it's going to be killed by the high pH, it's no good. I'm showing here a list of some of the most commonly researched bacteria. This includes the S. pasteuri, Bacillus sulfuricus, Bacillus megaterium, Bacillus subtilis, Bacillus cereus, and I do not know how to say that one, so I'm not going to say it. But all of these have been shown in literature to biomineralize successfully and survive in the concrete and provide all of those requirements that I mentioned on the previous page. We chose to use the Bacillus subtilis in this study because my collaborator Natalie Hull has done extensive work with this bacteria, so we knew a lot about it. We knew we had a good source of it, and we knew we could get it to do the things that we wanted to do and grow it to a sufficient size. Any of these bacteria will work, and I know the S. pasteuri has been used quite a bit. I'm showing also here the cost of this bacteria. So this is the amount that it costs to buy one sample of bacteria from a bacteria storehouse, essentially, that we have at Ohio State. And so if you want to grow bacteria, my original thought was this will be really expensive to get, but this is what you pay to get the initial sample. And then by using nutrient solutions, you can grow unlimited amounts of bacteria essentially forever, as long as you don't contaminate it or do something to kill it. So the costs are pretty minimal to get your sample of pure bacteria, and as long as you take care of it, it could work for years or decades. Or you could plan to use it for a year and then spend another $350 next year, which to me seems like not a huge amount to spend to make sure that your sample stays in good condition. But our other thought had been, there's been one study out of University of Ghent in Belgium where they looked at using impure systems. So what we purchased on the previous slide is this bottom picture where it says axenic culture. Axenic is a fancy biological term for pure. It's a single strain of bacteria. And in this previous work, they had used a multi-strain sample because axenic culture isn't what happens in nature. Never will we have a single bacteria growing in nature, and the presence of multiple types of organisms and bacteria within a system actually strengthens and makes that system more resilient because through natural selection, the weak ones die off and the strong ones stick around, and we naturally select for the most important properties. So our thought was, why would we want to just use this single system that in nature is set up to fail when we could get a non-axenic culture from our local environment here in Ohio for pretty much free, and then propagate that bacteria and have a more resilient system that maybe can last longer, maybe secretes more carbonate material, and overall works better and is cheaper for us. And indeed, that study that I mentioned from Ghent actually showed a reduction in cost from taking this method of about an order of magnitude. So they reduced their cost by 10 times through use of an impure culture rather than the pure Bacillus subtilis culture. So we wanted to investigate doing that here in Ohio with the thought of that we could lower our costs, increase the resilience of the system, and by taking samples in Ohio, maybe align the bacterial source with what it needs to survive in our specific Ohio environment, where we know it gets cold and not always the best weather. So we investigated a couple different non-extenic bacteria sources. The first thing that we did was we went to a bioenergy production facility, and I've shown a picture of what this kind of graphically looks like up in the upper right-hand corner of this slide. So in this facility, they accept food waste products, is what they told us. Here it's showing manure, but it will be similar by the time it arrives. So they accept oils and wasted food from fast food joints or other restaurants. They put it into a giant reactor that smells not very good, and the bacteria react and release methane gas. And so they send that to an electricity generator, burn it off, and create electricity from the bacteria decomposing in this tank. So we acquired a sample from them that would have been what's coming out of this effluent outlet here, and you can see a picture of the actual sample. It was straight up sludge, and it smelled really bad. And we actually had an accident in the lab where we spilled it all over a couple of things, and that was really gross. But so using this, we also investigated using distillery wastewater. I actually had wanted to work with some beer manufacturers to begin with. So I contacted Brewdog here in Columbus and also a Budweiser. And I think they thought it was kind of a joke that I wanted to take their material and withdraw bacteria from it and use it in concrete. I'm sure it's something that none of them have ever heard of. So I didn't get a lot of information back or willingness to work, but we did find a whiskey distillery in town who gave us the leftovers from their processes. Essentially, after they distill the spirit, then they're going to wash out their tanks, and that's full of the bacteria that performed the distillation, essentially, or the fermentation of the spirit. So you can see a picture there of our sample. This one wasn't sludgy and actually smelled really good, and was kind of fun to work with for a few weeks. Our last source that we got bacteria from is actually soil. And so a little bit of a spoiler, but we didn't find that either of these two types of bacteria worked very well, or were able to regrow well. And so we kind of went for soil as like a last ditch effort. I was looking at that list of bacteria and found that most of them naturally grow in the soil. So we thought, okay, with biomineralizing bacteria naturally in the soil, perhaps we can get that same type of bacteria out of our local soil. And so we dug up some topsoil from a garden in Ohio and took that into the lab. Okay, so here I'm showing on the graphs the number of cells at the initial 0 hour and after regrowing in solution for 24 hours for all three samples. The spent digestate from the bioethanol refinery is the SD. The DW is the distillery wastewater, and the soil is what's labeled soil. There's one more piece of information here is that the top is labeled with a V. Those are the vegetative cells, and then the S is the number of spores. While we do want to see high vegetative production, we also want to see the formation of spores, one, so that we know that that bacteria will actually form spores, and two, because we want to make sure that when it forms spores, they will survive. So if we don't see spores, that's going to indicate that this is not a good sample to use. So we tested all three of these sources at 2 pHs, pH 7, which is going to be a neutral friendly source that shouldn't kill the bacteria, and pH 9, which will be more analogous to using in our concrete. Obviously not completely the pH that we'll get to in the concrete, but if it can't survive in pH 9, it sure isn't going to survive in pH 12, 13, 14. Okay, so what we saw, we'll start first with the distillery wastewater. We essentially saw nothing from that. We were not able to grow any bacteria. We were not able to grow any spores in either the pH 7 or the pH 9 sample. This sample, when it arrived, was actually pH 3 or 4. So it was very acidic from the production of the whiskey. And so what we think happened is by increasing that pH to pH 7, we ended up killing off some acid-loving bacteria that were in the sample. But so sadly, despite this being a really fun solution to work with in the lab, it didn't work for our concrete system at all. So we had to move away from using distillery wastewater. The spent digestate was able to generate a large number of living vegetative cells. You can see we got up to 1 × 10⁸ number of cells after 24 hours. But both in the pH 7 and the pH 9 system, we grew no spores. So although we have living bacteria, we don't have spore-forming bacteria. So we're not going to be able to, again, use this successfully in our concrete system. In addition, Ohio State has some pretty strict safety rules regarding generation of bacteria. I think after what we've all experienced the last couple of years, we want to make sure that our labs that are researching pathogens and bacteria are really safe. And so I'm not complaining about this, but because they didn't know exactly what type of bacteria were in the sample, they were nervous to allow us to grow it in our concrete lab, and basically asked me to create an entire environmental chamber specifically just for doing this spent digestate growing work. So since we didn't see any spores, and since I couldn't afford to build an entire new lab to house this, we decided again to move away with that and move away from that, and we focused on the soil sample, which actually generated just as many living vegetative cells and also was able to form spores from our additional sample. So we have success with that. But two of the things that we need, living bacteria, growing bacteria, and spores forming. And we also saw that the spores and vegetative cells could exist at pH 9, which indicates that it might have some tolerance for higher pH as well. So from this point forward, we were working with the Bacillus subtilis axenic pure culture and the soil non-axenic kind of mixed bacteria culture. So the next process in the study was to evaluate the impact of different growth conditions. When we started this project, my original vision was that a concrete producer themselves would produce bacteria to go into the concrete. That's kind of migrated over time, and with some direction of our panel, to thinking more that this would be produced in a lab setting by an admixture producer such as SICA or BASF or GCP. So some of the things that we looked at may not be as relevant to a controlled lab situation, but we wanted to see how differences in concrete mixing labs might affect the growth and proliferation of the bacterial sample. So things we looked at, different pHs, pH 7 and pH 9, to see if the bacteria could grow and maybe survive better at higher pH. We looked at varying nutrient solution concentrations with the idea of reducing cost by lowering the amount of additional nutrient required, or maybe bolstering growth by increasing the total amount of nutrients provided to the bacteria. We also looked at varying temperatures with the idea that if you had an uncontrolled lab space, could you grow your bacteria in the summer? Could you grow it in the winter? Or did you have to condition that space to maintain a consistent temperature? We also investigated in stage 2 how to get the bacteria from the vegetative form into the spore form, and how to do that consistency and at high proportions. So we want to have the max amount of spores we can going into the concrete so that we have the max amount of bacteria surviving in the concrete and able to react and biomineralize. And then finally, we worked to confirm that they were actually biomineralizing in our sample and optimize those growth settings overall before moving into the lab for mortar and concrete testing. So I don't know about you, but in my concrete lab, we don't generally worry about germs and bacteria. I've told students before they need to use, when washing their dishware, things that remove ions from the surface of the glassware, calcium and alumina and things like that might contaminate our samples, but we're not working with anything that is living, so we don't really need soap. That is not the case at all with this work. So we want to make sure that everything is perfectly clean and decontaminated, so that we don't have something else coming in and eating away at our sample or taking over, especially the axenic sample. So to create the bacterial solutions, the first step is to autoclave everything. So we're going to heat everything at high temperature to kill it all off and to clean everything really well. We then get the aliquot, which is how the original bacteria source comes. When we made aliquots from the non-axenic culture, you essentially grow it up to high proportion, and then you centrifuge the bacteria out, and then you freeze it in these little pellets. I imagine this is pretty similar to if you take a probiotic, the same thing they're doing there. You get it in your capsule instead of in a liquid form. So we take that capsule and we'll inoculate that into a nutrient broth to wake the bacteria up and feed them and allow them to grow over time. We're going to incubate that solution at a specific temperature and shake it so that enough oxygen is available to feed the bacteria. And then we take the sample and we take 1 millimeter samples and put them on, we do what's called plating. Okay, so a plate is shown here on the bottom of the screen, and you can see they're numbered 0 to 7. At each plate, we are diluting the solution by an order of magnitude. So we're adding an additional 100 milliliters of water to each 1 milliliter sample. And with that, you can see that you get a diluted amount of bacteria. We start with a really dense pellet that you can't see through, and then it becomes a little bit less dense, and then they become kind of separated, and then you have a small amount. We do this so that we can actually count the bacteria. You dilute down until you can't see anymore, and we know that now that there's 10 to the 3 worth of bacteria in this sample. And you can see the little pen points here. You can count the number of colonies in each of these samples to determine what quantity of bacteria is alive in that sample. So clean everything, grow it over a certain amount of time at a certain temperature, and then plate it to count it. So the first step, we wanted to look at all of those variables and figure out what's going to allow us to grow the most bacteria in the shortest amount of time and for the cheapest cost. So our first bacteria we looked at, or our first variable we looked at, is time. We wanted to see how many days do I need to incubate this in order to get the maximum amount of bacteria. And this was really encouraging results. We see some variability within the first day of cooking those bacteria, and after 24 hours, it's pretty darn stable. So there's really no reason to grow the bacteria for more than 24 hours. Within a day, we can get to the maximum level. One thing I want to point out, so you may notice 10 to the 9th, if you've ever drank a kombucha or taken a probiotic, they often say on the bottle, 1 billion bacteria. That's essentially what we've created here, 10 to the 9 numbers of cell-forming units per milliliter equilibrates to 1 billion bacteria per milliliter of solution. So we're creating a very, very high number of cells with hopes that they'll survive over time. You might also notice that we've created orders of magnitude more numbers of cells in the soil sample compared to the B. subtilis sample. So this was initially very reassuring that non-extenic cultures, mixed cultures, were the way to go. We're already able to generate a lot more bacteria from that system compared to the pure system. The second variable we looked at was growth temperature. And primarily we wondered, does it matter what temperature my lab is, or can I grow these anytime? So we looked at 3 different temperatures, 10°C, 20, which would be a winter temperature, 25°C, which is about room temperature, and 37°C, which is about 100 degrees. And again, you can see we're looking both at the vegetative cells, which are shown with the solid lines, and the numbers of spores, which are shown in the dashed lines. The B. subtilis results are at the top of the page, and the soil sample results are at the bottom. So let's look first at the blue samples. Those are the low temperature samples. And what we saw with that was that we actually lost numbers of bacteria over 24 and 48 hours, and we were not able to generate very high numbers of bacteria at all. So we're about 4 orders of magnitude lower in the 10°C grown sample compared to 25 and 37°C. In addition, we created zero spores. So when we grow at low temperature, the bacteria really did not like that, and they either were growing so slowly that we couldn't track it over 48 hours, or they disliked it so much that they died. We saw a little bit different result from the soil sample. So with this, we did see by 48 hours, we could almost catch up to our other samples. But again, we had much lower spore formation. So in general, from this, we've learned it's not a good idea to try to grow your bacteria samples at low temperature. However, when we grew at higher temperatures, at room temperature and at summertime temperatures, with both of those, we had good cell formation. We had a decent number of spores at the end of the growth cycle. And with the soil, it was even more encouraging. We see increasing amounts of spores at higher temperatures. So to me, what this says is that if you're growing an actinic culture, if you're growing a pure bacteria, you shouldn't even incubate it to a hotter temperature. You can grow it in the summer, but you don't need to buy a high temperature environmental chamber to try and grow more bacteria. If you're growing a soil sample, however, you could benefit from use of a higher temperature system, because you can get orders of magnitudes higher spores from that approach. In order to create comparable systems, oh, hold on, that's on the next slide. All right, so third variable was pH. Our hypothesis here was perhaps if we grow bacteria at a higher pH, it will be able to live more successfully in a higher pH system at later time. We also wanted to see if one of them was more effective at growing at high pH or not. So again, we have the Bacillus subtilis sample at the top and the soil sample at the bottom. And what we see is that with the pure sample, we have a die-off of the bacteria in the first 24 hours when exposed to high temperature. By 48, it catches back up, but we did have a pretty significant drop in total number of cells in that first 24 hours. And again, we lost all our spores. So essentially, we cannot use higher pH system with the pure phase sample. We did not see that same behavior at all in the soil sample. So both the pH 7 and pH 9 had similar numbers of vegetative cells and similar numbers of spores. So this is confirming the hypothesis that the non-extenic mixed bacteria system is more resilient and more capable of surviving in challenging situations like within a concrete sample. Keep in mind, though, that these are in solutions with a bunch of nutrients, so they are not actually in concrete samples. So what we're seeing here is not reflective of what's in the concrete behavior. That will become apparent a little bit later. So for the sake of having similar test conditions and being able to compare bacteria samples, we decided at this point not to continue with pH 9 soil sample testing so that we could compare the pH 7 Bacillus subtilis and pH 7 soil conditions. So that is a place where I can see some future research that might be able to track better survivability of the non-extenic sample in concrete over time. We didn't include that in our testing here. Okay, so the last variable that we looked at was nutrient solution concentration. And so we looked at using a higher concentration solution to try and spur on additional cell formation. We also looked at using a half concentration solution to try and dilute the cost of the growth phase of this sample. Okay, so again, we have our vegetative cell growth in the top solid lines and our spore growth with the dashed lines, B subtilis at the top and soil sample at the bottom. So what we see with the Bacillus subtilis is that it doesn't matter what our solution concentration is, we get the same number of vegetative cells. And similarly, we get the same number of spores regardless of solution concentration. However, if we cook that Bacillus subtilis sample at a higher temperature, we can actually generate higher numbers of spores. And then similarly in the soil sample, we see pretty similar behavior across nutrient solution concentrations, a little bit higher spore count at the lower nutrient solution concentration. So this is good. We can actually generate more of the material we want with a lower cost solution. So after all of this testing, our final optimized growth recipe came out to be B subtilis at pH 7 at a higher temperature, so we can get those higher spore counts, and half nutrient solution concentration is what's typically recommended. And soil we can do at pH 7, room temperature, and again, the half nutrient solution concentration. Now for something completely different. We needed to, at this point, get those high vegetative cell counts, 10 to the 8, 10 to the 9 cells in our solution, get them all into spore form so that they can be then injected into the concrete. We're looking at two different methods of inducing sporulation in the samples. The way that we cause bacteria to sporulate and go from vegetative to spore form is to create a non-ideal system, a system it doesn't like and it feels uncomfortable in. That can be through use of non-ideal temperatures, through lack of nutrients or lack of moisture. That can be from an overload of minerals, essentially poisoning the bacteria, or that can be through changing the pH of the solution to one that the bacteria doesn't like. So our first approach was this mineral overload approach. We used what's called Schaefer's glucose solution, or 2XSG. And you can see it's a huge amount of different minerals, potassium chloride, magnesium sulfate, a bunch of calcium, iron, manganese, and then some glucose. We exposed our cells to that. The other approach was to try and dry the cells in the oven. So we're going to remove moisture and put them exposed to a high temperature that they don't like and see how many cells we get out of that. We essentially dried them to about an SSD condition, as much as you can tell that with a bacterial solution. And so what we saw, mineral solution results are on the left. We had decent sporulation, 10 to the 3rd numbers of cells per milliliter. And we're just looking at the green right now because we just care about the spores. But we saw a much increased numbers of spores through low temperature heat treatment. So we decided to go with heat treatment from here on out in order to maximize spore quantities. All right, our third step was to confirm that the bacteria can actually biomineralize. And we do this for two ways. Remember, there's two mechanisms. One is consumption of urea to create these carbonate materials that will interact with the calcium. We saw that the soil sample did this really well. It was able to consume a large amount of urea over the first 48 hours. Our soil, or our B subtilis sample, however, consumed basically nothing. So we were like, oh shoot, we just did all of this research, all this work, and we don't actually have a biomineralizing bacteria. But we tried again using thermogravimetric analysis, and you can see that we have peaks in the range where we would typically expect to see calcium carbonate for both of the samples, more in the soil sample, but also present in this B subtilis sample. And then we were also able to confirm the presence of calcium carbonate using XRD phase analysis as well. And you can also see here a picture of what these bacteria actually look like after we centrifuge them. So you can see we get a lot more volume of the soil sample for the same number of cells. And we had a lot clearer peaks in that sample, but we did still generate at least calcite and perhaps a little bit of vaterite, which is another form of calcium carbonate in our sample. So we were happy with that and decided to move on to concrete testing. And I was going to talk a little bit about what types of bacteria were actually in the sample, but it looks like we're running a little bit short on time, so I'm going to skip this. This is called microbial community. Basically, what we found is that we have something called Citrobacter frundi in the soil sample, and Ratutella orina, I don't even know how to say that. Both of these have been shown in research to be able to do things like remediate heavy metals in soils, biomineralize and repair cracks in previous research. So we think that that is what was present in our sample that's actually causing good results. Right, so our conclusions from the first phase of the work, we are able to generate a non-extending bacteria species that is capable of biomineralizing and forming spores. And so far, it looks like the soil is actually performing a lot better than that pure phase material. Right, so now what you've all been waiting for, laboratory, mortar, and concrete testing. So with this, we wanted to determine how mixture components affect the ability of the bacteria to live and also secrete calcium carbonate. So we wanted to look at things like SEMs, fly ash, which is an SEM, lightweight aggregate, if we need to use special water solutions to protect the bacteria at early ages, and a couple other variables. We wanted to track how long the bacteria was going to live in the concrete, how well it could heal cracks as well. Right, so one of the variables as well was curing methods. So I mentioned before, the typical method of doing this is ponding the samples in a nutrient solution. And we thought it would be a lot more feasible to go out and spray our concrete once a day and see if we could actually generate biomineralization with that approach instead. We also looked at mixing water solution. We used a phosphate buffer solution with the bacteria, which is kind of protective and gentle to the bacteria at early ages. We compared that with tap water to see if we need to use that PBS or if we can just go ahead with typical water systems. We also looked at whether we need to use lightweight aggregate or not, how fly ash affects it, and if use of an alternative CSA cement can improve the performance or not. We also investigated the effect of the environment and application of salt. Okay, so the process that we use to grow the bacteria is a little bit complicated. So we're essentially growing the bacteria for 24 or 48 hours. We then centrifuge the material and dry it at a high temperature, a medium temperature, 40 C in an oven. And then we can freeze that pellet until the moment we need to use it. Once we need to use it, we put everything back into a nutrient solution and then sorb that nutrient solution and bacteria into a lightweight aggregate, which is kind of like our probiotic pill that we're going to dose into the concrete. We added bacteria at a rate of 10 to 7 spores per cubic foot of concrete. And we did not use lightweight aggregate as the full fine aggregate amount. We only used 10% of the total fine aggregate in the lightweight aggregate. So this is a very small amount of material to add to your mix. And then we used dechlorinated water in all cases. So that could either be from well water or using water from the Columbus waterway. We allowed it to sit out for 24 hours in the lab. Okay, so after mixing it into the concrete, we tracked changes in cell concentrations over time. And so we're showing the B subtilis on the left and the soil sample on the right. And there's a couple things that you can see from this. First, there's no difference in performance between the OPC and the OPC using fly ash. So essentially, fly ash does not, despite having heavy metal content, does not harm the bacteria. And we can use this SEM without hesitation with this system. Secondly, we saw a lot higher numbers of bacteria when we used a calcium sulfoaluminate system. And we believe that's because calcium sulfoaluminate is known to have a lower pH pore solution, especially at early ages. And so that was able to kind of prevent killing off more bacteria in that system. So overall, more bacteria in CSA, no problem from use of fly ash. You also may notice that we do have some bacteria living all the way up till 90 days of testing. So hopefully up to 90 days after casting, we would still be able to get benefits from them. We also investigated the impact of the added bacteria on setting time and hydration kinetics. We see a little bit of delay in our isothermal calorimetry when we add bacteria into the mix, but we did not really see any significant change in setting time. So our initial set is pretty consistent across all the mixtures, but it does extend your final set time a little bit when you use the bacteria in an OPC system. When we added fly ash to the system, it seemed to kind of mitigate that final set effect. We had pretty consistent set times across all of the systems, bacteria or not. A little bit accelerated in the bacteria systems compared to fly ash. But it looks like, so this is OPC plus fly ash plus lightweight aggregate with no bacteria, and we also see that accelerated set time. So it may be the presence of the lightweight that's changed the setting a little bit compared to the bacteria. All right, so let's look at the impact of all these variables on bacteria survivability. First, we already mentioned, does the fly ash harm the bacteria? And the answer generally is no. So we see similar amounts of bacteria alive in both systems across the full testing time period. Secondly, does the lightweight aggregate improve bacteria survivability? This was pretty stark results. We see a big drop in our sample that did not have lightweights. No lightweight is the green at early ages, and that's maintained across the total timeline of the testing system. We see that similar drop a little bit later in the non-exenic in the soil system, but we still see a pretty stark difference between bacteria that's protected within that lightweight aggregate compared to bacteria that was just exposed straight away into the concrete with all of the mixing action and shear forces exposed to it. So it seems that it's pretty essential to use lightweight aggregate as a protection for the bacteria at early ages. So yes, we should use lightweight aggregate to improve survivability. Do we need to use a gentle curing solution, that phosphate buffer solution, or can we use tap water? So here we actually saw a reduced amount of bacteria alive at early ages with the phosphate buffer solution, and so we ended testing with that. So we actually see better performance of the bacteria using a tap water system. So no, we don't need to use a gentle curing solution. And then finally, does spraying or ponding the samples lead to better survivability? So in both cases, we see much more bacteria alive when we sprayed it compared to when we ponded in that curing solution. So by using the more feasible, easier once-a-day spray with nutrient solution, we actually keep our bacteria alive for a longer time. So spraying is better. You may be wondering what the effect of the bacteria on compressive strengths is. So I'm showing that here. The OPC system is shown in orange. The black is our fly ash OPC system, and then the blue is our calcium sulfoaluminate system. And the long and short answer is basically the bacteria don't affect our compressive strengths. We saw with the CSA system a slight increase, but it's still within the error bars on our measurement of compressive strength. So overall, there's no difference between samples with bacteria and without as far as strength goes, up to 90 days. Now what you've all been waiting for, the self-healing of the beams. So here I'm showing two flexural samples with fiber reinforcement to keep them from cracking quite all the way through. On the left is the pre-cracked picture. On the right is after, I believe, 6 weeks of curing. And you can see there is some pretty clear development of calcium carbonate in that crack region. It's not 100% through the crack. We found that it was limited to a certain size of crack. Above about 0.35 millimeters, we weren't able to get good crack healing. So if you have a fiber reinforced system that's going to hold those cracks together, or if you catch the cracking at very early ages, it seems that this bacteria concrete is able to heal that crack and fill it with calcium carbonate material. Wider cracks are going to take a longer time to heal or may not heal. So up to 8 weeks, we didn't see full closure, but we did obtain partial closure of the samples. We also did see increases in UPV, which is an indication of the total density of the concrete. And we also saw pretty significant drops in total water absorption when we tested sorptivity of the bacterial samples compared to the non-bacterial samples. So we do have evidence here that the crack and the pores of the concrete are being filled by calcium carbonate material and are doing what we had hoped they would. Here's a little bit of scanning electron microscopy imaging where you can see some indication of the bacteria. We see these kind of odd holes throughout the sample that are circular. Those indicate that a bacteria was at least there at some point and may now be dead. We also see the presence of some of the calcium carbonate crystals within the matrix, a little bit more obvious in the soil sample than in the OPC sample. We did also see increases in calcium carbonate quantities from shown in TGA. So remember we looked at this before, and calcium carbonate burns off between 6 and 800 C, and we see increasing area under the curve for the bacterial samples compared to the OPC. And you can see how that translates to as far as calcium carbonate percentage here on the right. So we're up to about a 12% calcium carbonate percentage in the B subtilis sample, a little bit lower at 8% in the soil, and down at 6% for our standard OPC. So we do see additional generation of materials with the presence of the bacteria. I was shown again, the next sample is the fly ash sample. We actually saw better crack healing with the B subtilis sample and the fly ash mixture. So again, the before and the after, we see almost complete crack healing, a little bit less significant in the soil sample. But in both samples, we do see, again, big drop in sorptivity and a big increase in ultrasonic pulse velocity, indicating densification of the matrix and healing of the sample. And again, you can see this one, you can actually see a bacteria in the sample in the microscope. And again, you can see the B subtilis over on the left-hand side. And again, we saw presence of higher calcium carbonate amount in the tested samples. And then finally, using the calcium sulfur aluminate mixtures, remember this is the one with much more bacteria alive in it over time. And we again see some pretty significant healing. Weird things happened in this one, and that it changed the color of the crack. It became really dark and almost wet looking, although it wasn't wet. But we can see that we pretty much can't see any of the crack available in either of the samples with the Bacillus subtilis or the soil sample. So crack healing worked really well with the CSA. It was a little bit prolonged. We cured this one for an additional 8 weeks to get to this point compared to the 6 in the OPC, and so that may be part of this as well. Again, we saw a little bit of an increase in UPV and a very significant drop in the amount of water absorbed. And so we're imagining a pretty significant reduction in porosity of these samples right in the cracked zone. And again, higher calcium carbonate amounts and evidence of bacteria from scanning electron microscopy. Okay. We also, as part of the concrete testing, performed a freeze-thaw durability testing. We wanted to see if the densification of the concrete would reduce overall freeze-thaw damage in samples. And unfortunately, we cast a QA or QC1 ODOT mixture, and it looks like that mixture is really high quality and does really well in freeze-thaw durability testing. So that's great. Yay, our standard mix works. We were not really able to tell a big difference between the bacteria samples and the control samples, though. We did see a little drop in dynamic modulus at the beginning of testing for the soil sample, but pretty much they both maintained 95+% durability factor over about double the amount of freeze-thaw cycles that are typically required. Usually you stop around 300, and we ran it for as long as we could, out to 500 cycles, and still really see just very durable concrete. So if you're using the system, you can count that you're not going to reduce your freeze-thaw durability through incorporation of the bacteria in any way. So I'm going to talk a little bit now about environmental condition testing. So we wanted to understand everything being done in the lab before. It's all been at room temperature, at very nice conditions, air-conditioned and heated buildings. That's not what our concrete experience is in the field, though. And so we wanted to know, how does the situation change when we're exposed to high temperatures, when we're exposed to low temperatures, when we salt the surface of our roads? And so we tested each of those conditions, and after or during the exposure to the temperatures and after the salt exposure, we sprayed curing solution on the samples for an additional 28 days to track changes in properties. With our hot weather testing, so hot is shown in yellow and cold is shown in green. We see increasing strength or increasing absorption actually with hotter temperature curing. This makes sense because we're essentially reducing the density of hydration products by accelerating the initial curing. And we did see, or we saw actually worse absorption in our bacteria samples compared to the control, which is in the solid material. Similarly, we see negative results from a cell count perspective as well, an immediate drop in numbers of cells that are alive in both the high temperature and low temperature curing samples, and pretty much a maintenance of low numbers of bacteria over time. If we're at 10 to the 1, that's like tens of bacteria alive per I think gram of cementitious paste that we tested. So very low numbers of bacteria actually survived the exposure to the temperature, which is unfortunate. And then as far as compressive strength goes, we saw initial increase in strength relative to the hot temperature. This is standard and what we would expect to see in regular OPC mixtures. So not really an effect of bacteria. And at later ages, we actually see a decrease in strength relative to higher temperature material. So it appears that overall the bacteria were not successful when exposed to hot or cold temperatures. They died off pretty easily. And didn't really generate any unexpected data from it. We also see with all of our crack testing, basically no crack healing. So again, the initial crack is on the left and the after curing crack is on the right, and we see almost no crack healing at all. So this is essentially really disappointing. And then, so we also tested our samples. We cured them for 28 days. Then we exposed them to a salt solution for 2 days. And then we cured them for another 28 days with curing solution, trying to track what was going on as far with absorption and cell counts and compressive strengths again. And so we see a reduction in absorption. So we actually had an improvement from the bacteria after exposure to salt, but we again saw a pretty steep drop in the number of bacteria that were alive after exposure to salt solution. So it's likely that this drop in absorption is due to crystallization of salt within the pores, not due to the bacteria doing anything to improve it. And kind of along those lines, we saw a drop in strength over time in the bacterial samples, or a maintenance of strength in the soil sample. So, and again, minimal crack healing in all of the samples. So overall, it looks like we need more work to understand the effect of temperature on bacteria and how to mitigate that, how to prolong the life of the bacteria when it's exposed to real concrete situations. Let's talk a little bit quickly about our concrete installation. We installed a 12-foot by 6-foot concrete pad near the Learning Gardens at Ohio State. If you ever come out and visit, there's a big garden area that you can walk through with all sorts of experimental stuff. It's really beautiful. Feel free to go and check this out. It'll be in place for at least a couple more months. You can see our mix design here with a pretty standard cement and fly ash content. It was 20% fly ash and about a 7-sack mix. And we did incorporate lightweight aggregate to protect the bacteria in it. When we cast this, we have 2 sets of control slabs too, because we wanted to track the influence of the curing solution itself on additional curing and changes in strength. And we also have a completely non-cured samples to try and see just what the OPC does by itself. Okay. And so again, I think we probably cast too high of quality concrete. We should have not gone for an ODOT specified mix, and we should have designed something with a lower cement content and was more prone to cracking, because after a year, the control did not crack at all. The bacteria slabs did not crack at all. So we're not seeing cracking in the bacterial slabs, which is good, but it essentially provides us not a lot of indication that anything was working. We had planned to count the number of bacteria alive in this pad over time, and all of our samples were contaminated, probably because people were walking across them with their dirty shoes or tracking soil where one of our bacteria came from across the pad, and it's sitting out in the environment getting rained on and washed over. So we essentially got numbers that just made no sense at all and didn't indicate that there was anything alive or dead. So we were unable to track the success of that. Overall, we saw not a lot of difference between bacteria samples and control samples as far as compressive strengths, resistivity, which we tried to track. It was a little bit higher in cured samples compared to the non-cured, but there's no indication that that was because of the bacteria. We also saw lower amounts of shrinkage in bacterial samples compared to the fully cured OPC sample, but more shrinkage than in the non-cured sample, which is a little weird. All right, and lastly, I'm sure you're all dying to know what the cost of this will be. So we tracked the costs of the growth solution, of the electricity required to incubate at higher temperatures, and the amount of solution we would need to grow in order to inoculate the concrete with a little bit more bacteria than we had initially put into it. And you can see the amount of materials per cubic yard of concrete are pretty high. So it's very it's very important to control the variables of this mix, otherwise it becomes just insane and doesn't make any sense to use in your system. However, if you do control and minimize temperature, minimize the amount of curing solution that you're using, we can get to a production cost of around $100, $130 per cubic yard. And then there were a few changes that were made to the concrete mixtures. We had to dechlorinate our water with a special chemical. We also had to incorporate the lightweight aggregate in the bacterial concrete. And so you can see the difference between a standard fly ash concrete and a concrete with bacteria in it is about a 10% increase. And then we also have to take into mind the amount of curing, and this doesn't factor in labor, but just the cost of the curing solution ends up being about $1.50 to $2 per square foot of crack healing you're trying to do. So overall, the cost ends up being about double in bacteria mixes compared to non-bacteria mixes. We did see a little bit of life cycle improvement in the samples when we used bacteria, but overall that doesn't account for a huge increase in life or construction costs. So we saw, modeling this with Life 365, a slight increase in the time to corrosion initiation, which would lead to a small decrease in repair costs over time. And so all in all, this will end up being about a 6% increase in your total costs over a 100-year construction period. Right, so things we learned from this, spray curing works better than ponding, which is good because that'll be cheaper and more implementable. Lightweight aggregate is essential for bacteria mixes. We can use tap water if we dechlorinate it. We can use fly ash and standard concrete mixes that we use in Ohio, and the CSA cement is pretty promising. Overall, the effect of the bacteria wasn't very significant in terms of compressive strength, but we did see reductions in sorptivity and improved UPV, indicating densified microstructure. We also saw that crack healing was possible, at least in our samples that were not exposed to high and low temperature and salt. We didn't really see any effect on shrinkage or freeze thaw. But the kind of scale of this system to real work was pretty disappointing. Non-ideal temperatures appeared to kind of mitigate any effects that we would see, as did salt exposure. And initial costs are quite high. So my recommendations are, if you want to use this, you might as well choose a pure material. The B subtilis had a little bit better results in the concrete than the soil sample did. but either one will work. You must incorporate lightweight into your mixes. And then ideally you would plan to place these mixes in spring or fall so that they have a little bit of time to exist in a kind of comfortable temperature range and prevent the bacterial cell death that could occur quickly. However, overall, I cannot recommend using this in your city or town or state roads at this time. I think a lot more work is needed to really understand how much bacteria we need in order to prolong the life of it and how to protect it from those temperature and salt exposures. All right, so I think I've talked way too long. And Paula, am I allowed to take questions still when we're at 2 o'clock?
Sure, go ahead. As long as the audience wants to stay on, go ahead and answer those questions.
Okay. Okay, someone corrected me about fermentation, that it is done from yeast. But I believe what happens is the bacteria consume the yeast and help to grow it. Dawn, you can correct me again if I'm still wrong. So one question is, what happens if we stop spraying curing solution? So what we've discovered, because we actually tested an additional set of samples where we just had a bacteria within the curing solution, we thought maybe we can just apply a bacteria to the sample after it cracks when we need it, rather than incorporating the bacteria through all this work at the beginning. And we actually didn't see any healing in that sample that just had bacteria in the curing solution. So the curing solution is necessary when the crack happens. You're essentially feeding the bacteria, and since the crack has happened, that's opened them up to oxygen as well. So they're breathing, they're getting the curing solution, that's enabling them to precipitate. I would say that you should stop spraying the curing solution once you see crack healing, or if after 4 or 6 weeks you don't see any crack healing, it's possible that the bacteria may be dead and not working. So you don't have to spray curing solution for the entire life of the bacteria. You just begin spraying it when the crack happens, and you can stop spraying it when you're happy with the amount of precipitation that's occurred within that crack. And those are the only two questions. If anyone else has questions, you're welcome, I think, to unmute and ask me or type it into the question panel.
Well, users can't unmute. All the questions have to go in that questions
Okay.
pod. But
I guess you can't talk to me.
Don responded.
Yeah, so he says there's no bacteria in the fermentation. Bacteria means a bad batch. All right, so well, I guess it's good that we didn't really grow any bacteria in those samples then.
And as of right now, there are no other questions. If anything does come up, we can, oh, we have one more. Hold on.
Oh, okay, so how long after the mixing did you wait for cracks? So those concrete slabs have been placed for about 1.5 years. They were placed in early June 2021. And so we've been waiting around for cracks for 1.5 years. I think part of the problem, let's see if I can go back to the picture of that. Part of the problem is that we didn't put any, we should have put something in the middle of these sections so that they're not connected together. I had planned out to have really long, skinny sections to try and induce additional cracking, but then we placed them right next to each other. And so I think it's a fairly square-ish section, which probably provided some restraint and kept it from cracking a little bit. So that's unfortunate and a lesson learned, and I'll try to do better getting my concrete to crack in the next go around. Great question. Anyone else have another question? How long do you expect the bacteria to survive in the concrete itself without any spray of nutrients? So what we saw in the lab testing was that we had, let's see, this one shows it. So look at the black, that we still have bacteria present up to 90 days. But I'm unsure that having tens or this may be hundreds of bacteria in the sample is enough to induce sporulation. And that's part of what I think more research needs to be done on is how many bacteria, what's the threshold below which nothing happens? Is it 10 to the 2, or do I need to maintain a 10,000 bacteria per gram of cementitious material in order to get good biomineralization. You also need to know what the dosage amount is to put into the concrete. All the literature before that we looked at was really unclear about how they were adding bacteria and in what proportion to the concrete. They said they added 10 to the 8th bacteria. So I think they took a solution with 10 to the 8th vegetative cells, and they didn't pay attention at all to the spore count, which is really what matters. So we need to know more about how much we need to put in, how much need to survive in order to actually cause the crack healing. But we performed the crack healing on the not hot or cold samples, and honestly on the hot and cold samples after 28 days. So it should have been in this range where we were seeing crack healing. So while we have bacteria alive at 90 days, I'm unsure that it's enough to heal the crack. So this, if you want to use this for crack healing, it would need to be for very early age autogenous shrinkage or drying shrinkage within the first 90 days for sure. So don't expect that you to put in a bacteria concrete and then 5 years down the road have a flexural crack form and heal with the bacteria. That right now to me seems unlikely. Let's see. Would the crack healing be noticeable without the microscope observation you showed? Oh, I should have said that. Thank you for this question. So these pictures are not microscope images. This is my student's iPhone in the lab. And so this is this is a sulfate mortar bar, a 1 inch by 1 inch by 11 inch mortar bar. And you can see almost the entire width of it here. So this is a 1 inch section we're showing the crack healing on. You can visibly see it with your eye in the lab. So it's not a microscope tracking thing. The microscope was here where we were trying to see if we could see bacteria, if we could see calcium carbonate actually forming in the paste. And yeah, I realize you're not going to do this in most labs, and it's a little bit excessive length to go to. But here you can very visibly with your eye see the crack healing. Let's see. How was the crack induced in the sample? So these were fiber reinforced mortar bars. And then we applied a small amount of strain to them to cause the crack in the bottom. And so you can actually see, we didn't test the bottom flange crack because they were a little bit too wide. It's pretty difficult to control exactly hit the crack width that you need. So we tested along the side of the beam, and you can actually see it gets wider at the top here than at the bottom. So we induced a crack because they were fiber reinforced. It kind of held them together and closed it up a little bit. And then we sprayed it with curing solution. So yeah, no, these were not shrinkage induced cracks. These were, we applied a force to the mortar bars in order to force it to crack. In none of our samples, including the shrinkage measurements I showed at the beginning, were we actually able to get the samples to crack. And so part of what I didn't say with this concrete, keep in mind that it has a bunch of lightweight aggregate in it. So that lightweight aggregate, and that's included in the control samples as well, because we wanted to mimic everything except the bacteria. The lightweight aggregate is known to be an internal curing agent, and so you pond it in water before you use it, and then it will slowly release water from the aggregate over time. So it essentially provides an exponential increase in curing. There's a lot of research out of from INDOT and Jason Weiss's group when he was at Purdue that looked at use of lightweight aggregate as internal curing agent. And it does a really great job at controlling curing or controlling cracking and minimizing cracking just through the presence of the lightweight aggregate. So that probably is also a big part of why we didn't achieve cracking in the concrete section. It was too high a quality. We used an internal curing agent, and we placed something that was relatively square. All of those, I think, probably contributed. Okay, so additional question is, so the cracks only developed in the mortar samples or the concrete samples as well? The cracks only developed in the mortar samples that we subjected to a flexural load. So they were only where we caused it to crack ourselves. So we really just wanted to test and see, like, could it actually feel the crack, or is this all like fake news, you know? And I don't think it is. It looks like they cured pretty well in the right conditions, but less evidence in the field.
We have no other questions at this time.
I appreciate those of you who sent me questions and can have a little bit of a conversation. If you have further interest, I'm told that the final report will be published in early December. So that'll be available to you. This is a really fun topic, and honestly, I love talking about it. So if you have other questions, feel free to reach out to me by email. My email is my last name, Burris, Burris .189@osu.edu. Or you can look me up on the OSU website too.
I'll also put that in the follow-up email for everyone that I'm going
Okay.
to send out. So Lisa, thank you so much for your time today, and audience, thank you for attending.
Thanks everyone for coming. I appreciate it.
Thank you.